Have you ever struggled to analyze ligand-receptor interactions in your spatial transcriptome data? If so, a powerful tool I'm going to show you today called SpatialDM may help you overcome this challenge. Techniques to study ligand-receptor interactions have been evolving. Affinity purification and mass spectrometry were very popular in the past, but you can imagine it's labor-intensive and low throughput. Since single-cell sequencing got prevalent, there have been a few high-throughput methods to identify LRI, ligand-receptor interactions, such as CellChat, SpatialDM, and SNI. For example, Ramachandran used SpatialDM to identify a signaling between macrophage subtype and mesenchymal subtype that could promote liver fibrosis. But there are a number of things that should be aware of for these methods. Number one, it'll be tough to match interaction to the original tissue, especially when the cell type is spread over tissue. Number two, you need to understand that these methods fundamentally rely on overexpressed genes of each annotated cell cluster. So if the two clusters are actually spatially distant from each other, it's very likely to be a false positive because most interactions take place in a distant range around 100 micrometers per micromole. On the other hand, it's often not the whole cluster of cells interacting, rather just the boundary between two subtypes interacting, shown in the pair B. So in this scenario, the cluster-based approach would neglect such cases. With the development of spatial transcriptome, we think such problems could be addressed, and that's what led to our project, SpatialDM. We get rid of all the clustering and annotation steps and observe each single spot directly to avoid false positives and false negatives. So how we do it? We screen a manually curated database around 2,000 pairs and adapt a geostatistic term, Moran's I, into a bivariate format called Moran's R to measure the spatial correlation between two variables, the ligand X and the receptor Y. So the higher the R, the higher the correlation between the ligand and receptor in a spatial context. To test this hypothesis, we can use the permutation modes like other methods, but we also derive a variance that could transform it into a z-score testing problem. So that's the global Moran to identify which pair of interaction exists in your data. And next, we have local Moran to identify at which spots the interaction takes place. So basically, if the high ligand expressing spots surrounded with high receptor expressing spots, the spot will have a higher local Moran R and vice versa. And similarly, you can apply these score testing to save your computational resources. We also included a differential analysis module based on generalized Miller model to test if a difference in condition is actually affecting your difference in global Moran statistics. And here we show many immune or inflammation-related interactions are enriched in adult colorectal cancer samples compared to healthy fetus samples. And we are thrilled by the speed of SpatialDM. It's capable of scaling up to 1 million spots in just 12 minutes using just one core. And this was made possible through our z-score testing option. And as mentioned, it gets rid of the arbitrary clustering and annotation. So it's expected to have low false positive and low false negative rates. And we tested using our simulated data. And indeed, only SpatialDM, a new method spatial core, have nice controls over false positive, shown on the left panel. The middle ROC panel shows our methods have a highest power in one simulated scenario, and it holds true across other simulated scenarios shown on the right panel. Now let me demonstrate how to apply SpatialDM in a real-world example. A melanoma dataset generated by a customized spatial transcriptome platform and has around 100 micrometers spot-to-spot distance. So here we refine the signaling distance to the nearest spots only. Of course, if you have a shorter spot-to-spot distance, that means higher resolution, you can make the signaling radius larger by adjusting these parameters. So Global Maran selected 103 pairs, highlighted in orange, including some well-known pairs in melanoma like CSF1, VEGF-A, and SPB1. Next, we used local Maran to identify the interacting hotspots for each selected interaction and could be visualized using our plotting module shown on the left panel. And you can see it has good concordance with the ligand and receptor expression shown on the right. And thanks to the SpatialDM module, we could cluster all the interactions based on their local selection spots. So here we have 3 patterns, lymphoid corresponding to three different cell types, lymphoid, melanoma, and boundary. Let's take a closer look at pattern 0, the lymphoid region, which is enriched with CD23 interactions. CD23 is encoded by FCER2 ligand and interact with receptors like CER2. And our course plot, also including our plotting modules, showed it's enriched with B cell and other immune cells, which is consistent with other as a single cell RNA dataset. And when we extracted the overexpressed genes of these CD23 hotspots and did geo-analysis, we found these spots to have an anti-tumor role, as opposed to some immune cells previously reported have pro-tumor roles. To wrap up, SpatialDM is a powerful tool that can help you identify co-expressed ligand receptor patterns in your spatial transcriptomics data, and it outperforms existing methods in power, false positive controls, and speed. And you can access our tool from our GitHub and apply to your end data objects seamlessly. I demonstrated the basic global and local selection functions today in the melanoma dataset, and showcase how these interaction patterns could be further extrapolated to some interesting biology questions by the CD23 example. We also included other dataset of different species, of different platforms, and of different size. You are welcome to take a look at our paper through this link, and especially this interesting differential analysis part for the intestine cancer dataset. And last but not least, I would like to thank many people for their support, especially my supervisors Yuanhua and Pantao, and my co-author Tianjie for deriving this score option. And yeah, my name is Zhuoxuan from University of Hong Kong. Welcome to DM me around my poster number 171 today, or through the happy hour. That's it. Thanks for your attention. I'm happy to take any questions.
Thank you very much. Do we have questions from our audience? Perhaps I would have a similar question for you, because in principle we are looking and focusing on responsible and reproducible research, and I was very happy to see that you provided also the GitHub, I think, right? So I saw that you work with melanoma, which is great basically, because there is kind of a heterogeneity and so on. But have you think if this could be applied to other types of cancer, just kind of like more solid, I would say? So I know that there is huge difference between liquid and solid cancer types, but I'm just curious.
Yeah, in principle, it can be applied all sorts of spatial transcriptome data, including all different types of cancer. And actually, we are pushing it in a very tiny part of our startup, which is for drug discovery, mainly for oncology diseases, including different kind of cancers. Yeah, and we're open for collaboration.
Thank you very much.